Well, good afternoon, everyone. I want to welcome you all to the Paul Marks Prize Symposium, both those of you in person as we're opening up again, as well as those on the Zoom link that we provided. When Paul Marks stepped down in 1999, after leading MSK for 20 years, the board decided to create the Paul Marks Prize for Cancer Research as a tribute to his contributions as both a leader and a mentor. The first Marks Prizes were awarded in 2001, and since that time have been awarded every other year in honor of outstanding young investigators in the field of cancer research. A lot of things have really changed since the last time we gathered to honor our Paul Marks Prize winners in 2019. The COVID pandemic changed much of the world. That's why we decided to meet both in person and virtually this year. And each of the award winners this year, of the 3 award winners, have agreed to give a separate lecture as part of the Presidential Research Seminar Series as we start up and re-engage ourselves as a community going forward. Today is the second of those events, and it recognizes one of our 3 2021 recipients. Their names were announced last November, but today we're honoring Sun He, who is the Oscar M. Schloss Professor of Harvard Medical School and Boston's Children's Hospital, as well as an investigator in the Howard Hughes Medical Institute. The other 2 2020 award winners, Ralph DeBerardinis and Charles Swanton, will be recognized separately. All 3 recipients are impressive in their contributions and the scope of their work. We are pleased, though, that Sun was able to join us in person to provide this second lecture, and she's joined by her husband, Jeff, and so we're very proud the family was able to come as well and join us as part of this event. It is important as we go forward in honoring this event, though, that we realize that something has changed in a major way since the last time we celebrated the Paul Marks Prize in 2019, and that is that we lost Paul Marks himself. Paul died in April of 2020 at the age of 93. Paul was a confident and visionary leader as well as a world-class scientist. Through his efforts, he helped set MSK on a pace on a more scientific course, and we're very proud of this prize in his honor. We also want to thank, before I do my formal introduction of Sun, that the prize selection committee is actually not composed of MSK scientists, but in fact scientists were gathered from the scientific community throughout the country. And this year was led by Celeste Simon at the Abramson Family Cancer Research Institute at the University of Pennsylvania, along with 5 other distinguished scientists, including a couple of prior Paul Marks winners. We thank them all for volunteering and their expertise and their time in making this year's selection such a great success. And now I'm pleased to introduce Sun to give her lecture. As I mentioned, she is both a professor at Harvard Medical School and a member of the Howard Hughes Medical Institute. Her laboratory studies how the innate immune system distinguishes between self and non-self nucleic acids. Her discovery has led to the understanding of the regulation of 2 key signaling pathways that play a role in immune responses and the pathogenesis of a wide variety of immune disorders. Her work also has important implications for today's forms of cancer immunotherapy. Sun earned her PhD degree from the University of California at Santa Barbara. She then went on to postdoc at UC San Francisco before joining Harvard and Boston Children's Hospital in 2008. As an independent scientist, she used her training in chemistry and structural biology to study a critical question in biology: how non-self nucleic acids are distinguished by a host cell to regulate innate immune responses. Her work has elucidated 2 key proteins, RIG-I and MDA5, in regulating cytoplasmic recognition of aberrant RNA. And it is my great pleasure to introduce her to give a lecture, an overview of her work, and recognition of the Paul Marks Prize. Sun, could you come forward and receive your award?
Thank you so much. Thank you. Hello, can you hear me? Okay. So thank you, Craig, for this generous introduction. I'm really truly honored to receive Paul Marks Award. I've never met Dr. Marks, unfortunately, but I read about his work on histone deacetylase inhibitor and the long list of his achievement as a leader who transformed Memorial Sloan Kettering Cancer Center. For that, I feel extremely privileged to receive this award that was made in honor of his life achievements. I want to take this opportunity to also thank others who have helped me throughout my career. There are many, of course, but I would like to start by thanking Dr. Fred Alt, who has nominated me for this award. Dr. Alt recruited me to Harvard Medical School as an assistant professor almost 14 years ago. And since then, he has been my colleague, mentor, and also a role model, not only as a scientist, but also as a leader who cares deeply about next generation scientists. And I'm definitely one of those people who benefited tremendously from his leadership. And I also want to thank both former and the current lab members who I've been extremely fortunate to have worked with. Without their dedication and hard work, I would not be standing here. I also want to thank my family, especially my husband, who is in the audience. For me, starting a new lab and running a lab was not always easy, and without his continued support and patience, none of this would have been possible. So I thank you for that. Now for the next 30 minutes or so, I'll give a brief introduction to what my lab has been working on, and that is about self-non-self discrimination in innate immunity. So as you know, all living organisms utilize let me change that to, okay, so I'm not entirely sure. Okay, so let me just, as you know, all living organisms utilize self-non-self discrimination as a fundamental basis for their defense against microbial infection. One of the most conserved ways for sensing viruses is to detect the viral RNA using a set of innate immune receptors. And in vertebrates, one important family of receptors that carry out this function are RIGI-like receptors. They play important central roles in antiviral defense because they are not only they are expressed in almost all tissues, and they also they activate the pathway, that immune response that is not just limited to the infected cells, but also can propagate to uninfected cells through the secreted mediator known as cytokines. Of course, this potent immune response is beneficial to the host only if self-non-self discrimination occurs accurately. And accumulating evidence from my lab and others suggests that misrecognition of self-RNA does occur, and this can lead to a constitutive activation of immune response and pathogenesis of a variety of different inflammatory disorder. Moreover, recent studies suggest that this same inflammatory pathway can actually a process can occur during a variety of different cancer therapies. And in this case, the pathway can play a beneficial role in by guiding the immune response against tumors. So RIGI-like receptor pathway have attracted much attention from a broad range, much attention from diverse fields, including host virus interaction, inflammatory disease, and cancer. My lab has been interested in understanding how the system works at the molecular level, both under normal and the pathologic conditions. So. And today I will talk about, I'll show you what we have found so far and some of our ongoing work towards the end. First, let me give you a brief introduction to RIGI and MDA5, RIGI-like receptors, RIGI and MDA5. These receptors were discovered by Takashi Fujita in 2004 and were found to function as in the cytoplasm as a soluble receptor. these receptors, and this immediately raised the important question as to how these receptors selectively recognize viral RNA against the high background of cellular RNA. While it was known that the double-strand RNA structure, duplex structure, are recognized by these receptors, it became also clear that the duplex length and modification and other chemical features also play an important role in this discrimination process. So this led to a deeper question of how these receptors really integrate this diverse and disparate features, information to make the self-non-self decision. So in order to, and at the time when I started my lab, the prevailing model in the field was that these receptors function as a simple switch that transition from the inactive state to the active state upon viral double-stranded RNA binding. At the same time, it was also believed that they tolerate self-RNA simply by not binding to cellular RNA. So, but this idea of simple switch really didn't, and affinity-based discrimination didn't really, couldn't really explain many observations at the time, and this is one of the observations that really got our attention. So in 2008, Shizuru Akira's lab published that MDA5 signaling or recognition requires a very long double-stranded RNA. In their experiment, it requires about 1 kb in order to activate MDA5, a kb double-stranded region in order to activate MDA5, and MDA5 signaling progressively increased with the length of the double-stranded RNA. I should mention that this threshold, length threshold, is not absolute, and this really depends on multiple factors, including the level of double-stranded RNA as well as the receptor itself. But generally it requires a several hundred base pair in order to activate MDA5 pathway. Now, this really, this length-dependent recognition or activation of MDA5 really made a biological sense and immediately, gave a clue as to how MDA5 can distinguish between self and non-self, because the double-stranded RNA that are generated during viral replication are often on the order of several thousand base pair, while cellular double-stranded tend to be much shorter. But to structural biologists and biochemists, this raises a really intriguing question as to how this small protein, MDA5, can measure the length of the RNA that is orders of magnitude bigger than itself. So in order to address this question, we first decided to look at how MDA5 binds double-stranded RNA and how that differs from its interaction with other types of nucleic acid, for example, single-stranded RNA or double-stranded DNA, which we knew do not activate MDA5. And this initial experiment immediately told us that there is something interesting. So these are native gel shift assay where you monitor the mobility shift of nucleic acid upon binding to protein, in this case MDA5. And what you can see here is that MDA5 binds to double-stranded RNA in an all or none fashion, and with a very little accumulation of intermediate species, even when there is an excess amount of double-stranded RNA, free RNA. And this is indicative, and this is a good indication that there is a highly cooperative binding. And this is in contrast to single-stranded RNA or double-stranded DNA binding. So MDA5, as you increase the amount of MDA5, you can see that there is a gradual shift from the low molecular weight complex to a high molecular weight complex, which is indicative of distributive and non-cooperative binding. So, in addition to this, we also found this is actually interesting. This gave an interesting idea how MDA5 may be able to distinguish between a variety of different nucleic acid. The way it binds is different, but the affinity was not that different. So these are, again, the non-stimulatory RNA, but with a very similar affinity, comparable affinity to double-stranded RNA, which are agonists on nucleic acid. Suggesting that affinity is really insufficient to explain the ligand selectivity of MDA5. So this really was the beginning of this finding really motivated us to embark on a journey that led us to identify and characterize a series of protein multimerization that is at the core of the regulated receptor pathway. In the next few slides, I will summarize very briefly the work that was done by a really talented group of postdocs and graduate students. First, we show that MDA5 forms a filament, and filament is formed by stacking this ring-shaped receptor molecule along the length of the double-stranded RNA. And we also found that this filament formation plays an important role in length measurement. So the filament stability is dependent on the length of the double-stranded RNA, and this is really the mechanism by which MDA5 regulates its signaling activity according to the length of the RNA. We also found that the related receptor RIGI forms a similar type of oligomer, filamentous oligomer, but the mechanism by which it forms a filament is completely different from the way MDA5 forms a filament. And this difference in the filament formation really allows RIGI and MDA5 to recognize distinct groups of viral RNA and function as non-redundant receptors for viral RNA. Now, even though there is a difference in the way they form filament and the type of RNA on which they form filament, once they form filament, the subsequent steps of RIGI and MDA5 activation process are very similar. So the filament, receptor filament, then recruits the cognate ubiquitin E3 ligases, Riplet for RIGI, TRIM65 for MDA5. And we found that this recruitment occurs in an avidity-dependent manner, allowing only the filamentous receptor to engage with the E3 ligase and move on to the next step of the pathway. And this E3 ligase then ubiquitinate the receptor, and at the same time, they bridge the filaments, forming this aggregate-like assemblies. And in this crowded environment, the signaling domain of the receptor forms a tetramer, which is then further stabilized by the ubiquitin chain that is conjugated by the E3 ligase. Now, this tetramer of the signaling domain then functions as a seed to nucleate yet another filament formation, and this time the downstream adapter molecule MABs. Now, MABs is a protein that is anchored to mitochondrial membrane, outer membrane, and I should also mention that the MABs filament was first discovered by James Chen at UT Southwestern. MABs filament then propagates efficiently on the surface of the mitochondria, and this then serves as a signaling platform to recruit and activate further downstream signaling molecules such as TRAFs and TBK1. Now, you can see that the pathway is very complicated, and this is clearly what we didn't really expect to find when we started this project. And the pathway really, even just between the receptor and the adapter, immediately downstream adapter, there are multiple steps of multimerization and the involvement of E3 ligase and ubiquitin chains. So you may wonder why this is so complicated. And this is a question that we have been asking ourselves quite a bit. And the way we think about this complexity is that maybe these individual steps may function as a checkpoint to block the, to filter out the majority of self-RNA and only allow non-self RNA to pass through and activate MABs. Once MABs is activated and nucleated, MABs filament can propagate very efficiently, allowing efficient amplification of the immune signal. So this multi-step assembly, we believe, is the key mechanism for ensuring both the accuracy and robustness of RNA discrimination. And we have some evidence that support this filter model, and we are currently following up to further develop this model. Now, even with these multiple checkpoints in place, the system is not fail-proof. And it turned out that the first step of a ligand receptor engagement is particularly vulnerable to errors. And when this step goes awry, the error can propagate to the rest of the pathway and cause a variety of different pathologies. And one of the first clues came from the work of my collaborator, Yannick Crow, who identified several pediatric patients with a de novo mutation in the MDA5 gene. He found that these patients show symptoms of severe inflammation, oftentimes in the brain, as you can see from the brain calcification, which closely mimics congenital viral infection. But the disease can affect the other tissues as well, such as skin and joints, and in a way that mimics symptoms of chilblain, lupus, and rheumatoid arthritis. We later found that these patient mutations are gain-of-function mutations that constitutively activate the receptor signaling activity. We also found that these mutations don't just spontaneously activate the pathway or the receptor signaling activity, but it still requires the binding to RNA, meaning that the receptor is constitutively active in these patients because they misrecognize, the mutants misrecognize self-RNA. So this raised a question: what kind of RNA, self-RNA, are responsible for the aberrant activation of MDA5 in the presence of gain-of-function mutation? So this question was actually, was really important, and we wanted to identify the RNA that, activate the receptor for a long time. And you may say, well, why don't you pull down MDA5 and see what RNA is associated? And this is obviously the approach that we took in the beginning. It turned out that this approach doesn't really work so well. And when you think about how MDA5 recognize RNA, this actually became clear that this approach is not appropriate. You may recall that the MDA5 can bind to a lot of different nucleic acids with a comparable affinity. With a single-strand RNA and double-strand RNA, we didn't really see any a clear difference in affinity. And the way they MDA5 distinguish between these nucleic acids is really by forming a filament on double-strand RNA. So on double-strand RNA, MDA5 forms filament and activates signaling. On single-strand RNA, it doesn't form filament and does not activate signaling. So when you pull down MDA5, you can imagine there will be a lot of nucleic acid, single-strand RNA that are coming along. And this is particularly problematic when you think about the fact that the RNA-seq approach usually, requires the enrichment of ligand. And it becomes even more problem when you think about this ratio of the ligand-receptor interaction. So for a given number of MDA5 molecule that you pull down, you're going to pull down a lot more single-strand RNA that are sparsely bound by the receptor compared to the double-strand, a few double-strand RNA that are coated with MDA5. So it became clear to us that we cannot use this kind of affinity-based approach because MDA5 does not utilize affinity to distinguish between self and non-self. And we must use the filament conformation-specific approach to identify the ligand. So it took a while for us to really figure out what approach to use to identify the ligand. And eventually, one method that really worked well was this RNase footprint assay. The experiment was fairly simple. You isolate cytosolic RNA, and then you incubate with MDA5. You're going to form a variety of different complexes, both filament and non-filament. Now you digest all the RNA with a ribonuclease A to the point that non-protected, unbound RNA will be completely digested. And now you will get a filamentous footprint for double-strand RNA and monomeric footprint for single-strand RNA. Now you eliminate the single-strand, the monomeric footprint by size selection and perform RNA-seq. RNA-seq result really showed that the result was actually quite striking. There was only one species, RNA species, that was enriched by this footprint assay, and that was ALU retroelement. Before digestion, where the footprint assay was 2 to 3%, and that became 70% after ribonuclease footprint assay. And it was not just any ALUs. So you can see that the ALUs that were enriched were almost always pairs of ALUs that were in inverted repeat configuration. This was really satisfying to us because inverted repeat ALUs were previously known to form this kind of a duplex, fold back and form this long duplex that is long enough to support MDA5 filament formation. I should also mention that these ALU RNAs are fairly common in the cytoplasm. In our estimation, about 1 to 2% of the RNAs are inverted repeat ALUs that form this kind of that can form this kind of double-strand RNA, which makes 300 base pairs sufficient to activate MDA5. Now, ALUs are very common, and this is actually not is ubiquitous as well. So they are often in the form of a 3' UTR in many different coding RNA. So it raises another question: why then wild-type MDA5 is not activated by ALU? Why are we walking around without the severe inflammation that is caused by MDA5 activation? And the answer turned out to be due to the nature of the ALU duplex RNA. So we found that the ALU duplex RNA are not the most optimal. It's a suboptimal RNA, duplex RNA. It often has bulges and mismatches and RNA modification, which interfere with the wild-type MDA5 filament formation and thus downstream signaling. Now, in the presence of gain-of-function mutation, the mutation actually increased MDA5's ability to form filament even on this imperfect duplex RNA, allowing MDA5 to form filament on these ALU duplex RNA and activate the pathway in a constitutive manner. We also found that the aberrant activation of MDA5 can occur even without a mutation in this receptor. When there is a loss-of-function mutation in an RNA-modifying enzyme, ADAR1, the lack of RNA modification leads to global increase in duplex structural integrity, and that leads to even the wild-type MDA5 to form filaments on inverted repeat ALU and activate the pathway and cause the same type of inflammatory disease caused by the gain-of-function MDA5. Now, misrecognition of self-RNA does not always lead to pathogenesis of inflammatory disease. And when this misrecognition occurs in a controlled manner, this can benefit the host as well. What these 3 studies showed earlier is that this can happen in the context of cancer therapies. What they found is that the 5-azacitidine which is the DNA methyltransferase inhibitor, and also the common chemotherapeutic agent. This reagent basically activate or de-repress a subset of RNA, including inverted repeat ALUs, and this leads to even a further increase in the level of ALU duplex RNA in the cell that leads to the activation of MDA5 and downstream interferon signaling pathway. What they also showed is that it synergizes, this antiviral signaling activity synergizes with the subsequent checkpoint immunotherapy for cancer suppression and plays an important role in the final outcome of the patients. And similarly, studies showed that the ionizing radiation therapy also leads to the activation of a similar pathway, and this time primarily RIG-I. So this was actually really interesting. It was previously thought that the genotoxic stress primarily lead to the activation of the DNA sensing pathway mediated by CGAS through the formation of the micronuclei, but these studies clearly show that a RIG-I pathway is activated during the response, depending on the cell, and that is also playing an important role in anti-cancer therapy. And in my mind, what is unclear so far is the exact identity of the RNA that is produced during this genotoxic stress and activate RIG-I, and we are currently working to understand the mechanism of the the RIG-I activation during genotoxic stress. So together, I think all of these data really suggests that the difference between self and non-self RNA is not black and white, and rather it is really the immune system that has evolutionarily optimized its threshold to avoid the recognition of the leaky levels of immune stimulatory RNA from the endogenous sources. The threshold as I mentioned, not defined by just a ligand receptor affinity, but rather it is determined by multiple steps of multimerization, both by the receptor, adapter, and the downstream pathway, and which all work together to fine-tune the threshold to avoid the self-RNA recognition. And the balance between this immunity and tolerance relationship breaks down when the threshold level is lower. For example, by having a gain of function. For example, in the case of a gain of function mutation in the receptor, or when the overall the level of the self-RNA, immune stimulatory self-RNA, is perturbed. For example, during cancer therapies. In both cases, there's a breaching of the self-tolerance, and that leads to the activation of the immune response. So some of our current work is really to understand more detail how the threshold level is determined, but also identify a small molecule that can really modulate the threshold level to either activate the immune response in a very selective way in the context of cancer therapy, or inhibit the immune response in the context of inflammatory disease. Now, in the remaining 10 minutes or so, I would like to share some of our ongoing work that we're very excited about. This is really related to how cells control kinetics and magnitude of immune response to avoid excessive and harmful inflammation. And this work was done by two very talented graduate students, Chris Cadena and Max Paget. So when you introduce double-stranded RNA to cells, one of the most prominent features that you're going to see is the formation of stress granules. This foci, cytoplasmic foci, known as stress granule. And this can be visualized by staining the cells for the stress granule marker G3BP. We became interested in stress granule because in 2012, Takashi Fujita's group showed that the stress granule recruit the receptor, RIG-I and MDA5. And we found that there is not just the receptor that are recruited, downstream adapter MABs and other signaling molecules are also recruited to stress granule. And this raised the obvious question, what's the role of stress granule in antiviral signaling? There have been several models that, suggest different aspects of the regulation, but one of the most prominent proposal is that the stress granule actually function as a signaling scaffold to further amplify the immune signal. And this sort of made sense to us at the time because the pathway, this pathway really works by multimerization at multiple levels. So if you concentrate the receptor and signaling molecules within the stress granule, there may be an amplification of immune response. So we decided to look further into the relationship between stress granule and the pathway. And before I go further, let me introduce a little bit about stress granule. Stress granules are non-membrane-bound molecular condensates formed by cytoplasmic RNA protein complexes. They are formed by a variety of different stress, including double-stranded RNA, nutrient starvation, ER stress, and so forth. They are sensed by different kinases, but the downstream effect is very similar. They all phosphorylate the translation initiation factor EIF2 alpha that leads to a global shutdown of protein synthesis, leading to the shift of the protein or ribosome mRNA complex from the polysome to the stalled initiation complex. Now, this stalled initiation complex has a long naked stretches of RNA, and this then aggregate together with many other RNA binding protein to form stress granule. So the whole process is called integrated stress response, and the physiological role of this response is well known. That is, by shutting down the protein synthesis of the majority of the genes, you save and preserve the energy and resources, and focus is cellular resource and energy for synthesizing genes that are essential for stress recovery, which most of which actually bypass this translational regulatory mechanism. So the integrated stress response, in particular the translational shutdown, is well understood, but exactly what function stress granule actually play is actually very controversial. This is not really required for translational shutdown. So the question that we became interested in was how the stress granule actually what role stress granule have in the innate immune signaling. And the way we are addressing this or approaching this is using in order to selectively alter stress granule without altering the upstream integrated stress response, we use this genetic tools, knockout of nucleators, some of the nucleator G3BPs or UBAP2L to selectively downregulate or suppress stress granule formation. Now, when we actually look at the regard-like receptor signaling, we found that these are actually stress granules are actually a stress granule suppress regard-like receptor signaling. These are the genes that are normally induced by double-stranded through the MAPS pathway, regard-like receptor pathway, and most of them are actually hyper-induced in the absence of stress granule. And we then use a variety of different biochemical approach to further confirm that the signaling pathway is activated, not just at the mRNA level, but also at the pathway level. And we were also further able to narrow down to one particular step that is actually hyper-activated in the absence of stress granule, and that was actually the MAPS step. I don't have data to show you, but the part that was really perturbed seemed to be mitochondria. We're currently looking to understand how mitochondrial changes in the mitochondrial homeostasis is affecting this, and how stress granule is affecting the mitochondrial homeostasis. Now, together, in addition to the innate immune signaling that is hyper-induced in the absence of stress granule. Another thing that we found was that the stress granule deficient cells undergo more prominent cell death upon double-stranded stimulation. The cells are actually perfectly fine in the absence of any double-stranded stimulation, but upon double-stranded stimulation, you see most of the cells are detached from the surface, and you can actually see that from the caspase activation or the cytox staining. This actually happens at all levels, all doses of double-stranded RNA, and the cell death occurs over the course of 24 hours. And we found that this cell death in stress granule deficient cell is apoptosis, and this is actually also specific to the type of stimuli. So if you apply other types of apoptotic stimuli, such as etoposide and staurosporine, you don't actually see the enhanced apoptosis in stress granule deficient cell. So there is something about double-stranded stimulation and the stress granule formation that really synergize to actually lead to the cell death when there is lack of stress granule formation. We found that this is primarily dependent on regard-like receptor pathway. So when you delete MAPS, you actually rescue most of the cell death. And we were further able to narrow down to a TNF-alpha pathway. This was actually interesting. So you can actually see that the TNF-alpha is more upregulated in the absence of stress granule, and anti-TNF-alpha actually leads to substantial reduction in cell death. This was interesting because it was completely independent of the interferon signaling pathway, and we show that this pathway does not play a role in cell death in the stress granule deficient cell because there is a built-in negative feedback loop. So you get a very transiently activated interferon signaling, but that is then suppressed very rapidly through this negative feedback mechanism. And the similar negative feedback doesn't seem to apply in other pathways, such as the one that controls the TNF-alpha expression. Now, to summarize the work that I've shown you, what we found is that the stress granules are really a potent suppressor of the immune response. And the way it negatively regulates the immune signaling is very interesting. It's not a feedback negative regulation, but instead it really controls the ramp-up speed of the immune signal. I didn't have time to actually go through some of the data, but it really controls the ramp-up speed of regard-like receptor signaling pathway, and that allows cells to keep the immune response under the death threshold and allows a sustained immune reaction. In the absence of stress granule, double-stranded stimulation leads to uncontrolled amplification of immune signaling, and that leads to the the hyperinduction of a variety of different pathways downstream of MAPS. IRF3 pathway seems to have a built-in negative feedback loop that leads to a rapid control. But other pathways, such as the one that produces TNF-alpha, doesn't have a similar negative feedback loop that leads to the hyperactivation and cell death, consequent cell death. So we think that the stress granules are not a signaling platform, but instead a buffer. It may function as a buffer or a shock absorber to prevent excessive immune response. We're not so sure what is absorbing at this point, and as I mentioned, we think that it has something to do with the mitochondrial metabolism or other homeostasis maintenance factor, and we are really looking forward to understand this connection between the mitochondria and immune signaling and stress granule in the future. With that, I would like to thank, again, all my lab members, both the current members and the past ones, and also all my collaborators. I feel truly fortunate to have had work with all of them, and want to thank you all for helping me become a better scientist. And thank you for your attention.
Well, I want to thank Sun for a very stimulating talk. I'm going to use the microphone here to tell people in the room there are 2 microphones to come up and ask, so please come up to the microphone. But in addition, Steve Long is here to help us look at questions from those of us that are on Zoom. So could you please enter into the Zoom queries, in the chat, the question, and Steve will call them out from here. But with that, we'll go to our first question.
Hi, Sun. It's a beautiful talk. So the MDA5 recognition of the ALU sequences, it's almost like, and you were saying it's an instructional role, right? It's co-opting the system of ALU sequences in humans. So in rodents, there are no ALU sequences.
Right.
So so in orthologs, is the recognition different, or is
right? So the great question. So yes, ALUs are primate specific. So your question is, what happened to rodents if you have? If you make the same mutation in mouse, then you also get the autoinflammation, and we think that that's due to B2, very similar B2 sign element. So ALU is a sign element in mouse rodents. There is an analogous sign element called B2. They are known to form double-stranded RNA. We haven't proven it but that's sort of the candidate RNA that probably play a role in mouse inflammation.
But this is it's a beautiful model. So basically there's a pressure based on some endogenous RNA to evolve the recognition. Mm-hmm. So it'll change with different.
Mm-hmm. Right. Yeah. So I think that our theory is a complete speculation right now. So rodent has B2, and they tend to be, if I remember correctly, I think B2 tend to be more abundant, or they actually tend. Let me try to think. I think that they. I haven't thought about it, but that was a long time ago. I think that there is a different level of complementarity compared to ALUs, and we suspect that the MDA5 sensitivity in rodents and human primate may be different because the level of and the type of double-stranded in the cell are different.
Very nice, doctor. Thank you.
So I was really interested with your TNF-alpha response to give cell death. And one other interesting thing, since obviously my lab studies bioenergetics, is your RIG-A multimerization is ATP dependent. Mm-hmm, mm-hmm. And so there's been lots of reports that TNF-alpha alters either the ability of mitochondria to produce ATP or to stimulate autophagy. So have you looked at whether or not this is about ATP levels and the ability to multimerize the RIG-I or MDA5 pathway in terms of the TNF effect?
Oh, I see. So the way I thought about it was, so we haven't looked at it from that perspective yet. So I didn't think about the effect of TNF-alpha back to the upstream process so far. But the part that we know is that in this pathway, we looked at all the steps, and the part that is affected by the stress granule was really the MAPS polymerization, or its ability to recruit the downstream signaling molecules. So we think that it is probably related to, not necessarily, it could be related to ATP, but we think it has something to do with the mitochondrial homeostasis. But we are looking into that right now.
Thanks. Eric Lai asked a question about dicer and if that's involved in restricting the double-stranded RNA population in the cell and if that's in any way modulated in pathogenic conditions.
Mm-hmm. Yeah. So dicer pathway and the interferon signaling pathway are known to have yin and yang relationship. So when there is interferon signaling, dicer is actually suppressed through a multiple different mechanisms. So whether dicer, and also obviously dicer has a suppressive effect on the MDA5 and RIG-I pathway by degrading the double-stranded RNA. But whether dicer plays a role in the inflammatory disease or this auto-inflammatory condition is something that I think there is no genetic evidence yet, but it would be really interesting to look further into that.
Is there a directionality to the filament formation? Does it move from one end to the other?
Yeah, it does,
okay.
Actually. So ATP hydrolysis, so I didn't talk about it at all, but these filaments are not really static filaments. So they go through assembly, and especially MDA5, they go through assembly and disassembly process in an ATP-dependent manner. And we think that it is directional. We actually couldn't figure out exactly which direction it's going. So because of this dynamic instability, there is a collective movement of the filament along the length of the RNA. And we actually found that also contribute to some of the remodeling activity. So some of the RNP complexes can be remodeled by the filament dynamics.
Hi, Sun. I really enjoyed that talk. I was curious to understand better the mechanism by which the stress granules were seem to be, I guess, was suppressing the activation of MAVs. Is it just a physical sequestration of proteins that otherwise would, can go around and identify double-stranded RNA better, or is there something more complicated?
So that was
happening?
our initial hypothesis, maybe because we see, by imaging MAVs foci really perfectly co-localized with stress granule. We thought that there's a stress granule sequestering the signaling molecule. So that was the initial hypothesis. But we don't see the evidence. I think that there's a very small fraction of MAVs that is actually localizing with stress granule. Somehow we see mitochondrial change all over the cell. And we don't know what the connection is, exactly what molecule is responsible for this crosstalk between mitochondria and the stress granule. And yeah, so we don't know the answer, but we know it's not the sequestration mechanism.
Okay, and the early filaments that form, are those happening in a specific places within the cell, or is that just sort of all throughout the cytosol?
Yeah, we don't know. So as soon as you introduce double-stranded RNA, there's a stress granule formation. And actually stress granule formation and the recruitment of MDA5 molecule to stress granule is independent of its activity, signaling activity. Where is the active filament? We don't actually know that. It's something that we are kind of working on. Hi, Saishan.
Hey, San. Great talk. Do you know whether response sensing by, let's say, toll-like receptors is affected in the absence of when you disrupt stress granules genetically?
We haven't looked at that. It would be interesting. We did look at C-gas pathway, which doesn't involve stress granule formation. So the reason that we ask is whether the downstream pathway, just general immune signaling, is affected. We didn't see that. So whether toll-like receptor, do you have any particular reason to suspect toll-like receptor?
Well, you can sort of look at the sensing of the same sort of ligand being double-stranded RNA by toll-like receptors. Perhaps would be a good control for a specific role for stress granules in RIG-I MDA5 pathway.
So the cells that we have been using, a lot of them are cancer cell lines, but they don't have a toll-like receptor 3 active. So I think that we have genetically tested all the immune response that we see are completely RIG-I receptor dependent, actually particularly RIG-I. based on the.
Well, that would be
Yeah, but
that would require transfer.
right. Yeah, that would be something to look into.
That was a great body of work that you presented, so thank you for that. There is an evolving theme that the innate immunity activation pathways are different between early stage carcinoma cells, stage 1, and later stage metastatic carcinoma cells that have already been through a number of stresses and made it through. I know the STING pathway, for example. The STING pathway is active in a canonical way in early stage cancer cells, but it's twisted and ineffective, in fact, in more advanced ones. Do you think that the response to 5-ASA, for example, is going to be different between early stage and later stage carcinoma cells?
You mean this pathway, whether?
Yes.
So yeah, I've looked at some of those papers where STING pathway sort of gradually loses its activity over the course of cancer development. People found that that is not necessarily the case for RIG-I receptors. And I think that when people looked at a variety of different cell lines, cancer cell lines, the STING pathway is a lot of the cell lines lose the STING pathway, but RIG-I receptor pathway are actually active. So most of the cell line, if you look at it, RIG-I receptor pathways are active. I should actually say RIG-I pathway is active. MDA5 pathways, most of the cases, is dead. So somehow, I think that the cancer sort of found that the RIG-I pathway is okay to keep, but the STING and MDA5 pathway is not okay. I don't know what's the explanation for that. Maybe it has something to do with endogenous level of ligands that can normally trigger MDA5 and STING or CGAS STING. Whereas RIG-I, I think that there is a much higher bar, and so if you do some kind of a very crude experiment, you can see that the RIG-I doesn't really have a whole lot of endogenous ligand in the cell in the normal condition. So maybe that's the reason, and that might be the explanation, but I don't have any particular explanation beyond that.
All right, thanks.
Okay, a couple more questions from the chat. Vladimir Despik asks, it is known that the lack of the 2' O-methylation on the first nucleotide of endogenous transcripts causes robust RIG-I activation. Are you familiar with that?
Lack of 3'?
Lack of a 2' O-methylation.
Oh, yeah.
And then how do you think the mRNA in this case causes RIG-I activation, given that they are not fully double-stranded on their 5' end?
So we don't know. As I mentioned, the RIG-I activation and the ligand, endogenous ligand, to me is still mysterious. So that's something that we are looking at. Whether it's really the mRNA that lacks the 2' O-methylation, we don't know. I think you need to have some kind of structure. So you can't just have a 5' triphosphate or even 7-methylguanosine cap. You do need to have a structure in order to activate. And the exact ligand for RIG-I activation is not clear. And I should also mention that what I'm showing you here is really largely based on the perfect double-stranded RNA that we introduced to the cell. But in cell, I think that there are many RNA that with a complex RNA structure. With RIG-I, I think that those complex structures will play a role, and maybe the complex that will form with RIG-I may not be the beautiful filament or simple filament, but maybe more different three-dimensional structure. That's something that we are currently working on.
Great. And then there was another question about methylation of RNA being partitioned to the stress granules and what effect that might have on, do they need to be removed, for example, or how does that affect the process?
I am actually less familiar with that story, and what we know is that the double-stranded RNA, the one that is activating the pathway, they do not go to stress granule. So that's all we know. So the stress granule is not the signaling location, it's doing something else. As I mentioned, we are still trying to figure out what it exactly does.
I had a question too, which was, do you think there could be any other cellular factors that might depolymerize or remove some subunits or affect the balance of these?
So especially the MAVs, this is still really mysterious to me. The MAV filaments are formed. Obviously, immune signaling goes up innate immune signal, the interferon pathway goes up and down. MAPS level don't really change a whole lot. So we think that there is some mechanism that is filament is disintegrated or dissolved. We don't know exactly what happens. It's a very controversial. Yeah, I can talk about that more, but yeah, there is something in there, but in the cell that controls the polymerization process and depolymerization process, but we don't fully understand.
All right, well, let's join together in thanking Dr. He for a lovely seminar.
Thank you.